So welcome to the first week, the part two of the lecture series for the week. Today we'll be talking about some of the different next generation sequencing applications. As bioinformaticians, in order to do good high quality analysis, oftentimes we really need to understand sort of the goals of the various sequencing projects. So we'll talk a little bit about that today. So I kind of came up with basically three different areas in which next generation sequencing is being applied. One area is in sort of what we all know is sequence-based analysis. What is the sequence of a genome? What are the genotypes of individuals at individual genome positions? And, in some cases we want to know the different mutations that have appeared in cancerous tissues, and so we can sequence a cancer or a tumor cell and compare that back to the somatic tissues of an individual. So I'm including in this sort of the epigenome sequencing, what we call resequencing, which is the sequencing of populations of individuals. Second area of applications is sort of, broadly speaking, is quantification. So here all of you know about RNA-seq, and here what we're interested in doing is quantifying the number of molecules in a particular preparation, such as a cell or a cell under a certain set of conditions. Also in this category is what we call ChIP-seq. This is chromatin immunoprecipitation and sequencing. We'll be going into that in some detail, and also we're interested sometimes in just genome sequence. We're interested in copy number variation, so we are wanting to quantify the amount of DNA in an individual in a region that may differ in copy number among individuals. And finally, paired-end sequencing in particular is used for genome assembly or transcriptome assembly. So oftentimes we can de novo assemble a piece of DNA, but we don't know which contigs or the products of that assembly are associated with other contigs. We can use paired-end sequencing to help with that. We can also help to identify alternative splice variants. These are mRNA molecules that contain different exon complements. I think I mentioned that in the last lecture. And finally, we can hope to detect structural variations. These are translocations and other forms of chromosomal rearrangements. So many of you probably know what a SNP is. These are single nucleotide polymorphisms. This is probably one of the basic goals of what we call whole genome resequencing. I'm highlighting re here because the idea of resequencing conceptually is that these are genomes that this isn't the first individual of a species that is sequenced. Remember that we have these reference genomes, which oftentimes reflect the first individual, but resequencing is we're sequencing again and again and again different genotypes of the same species, which presumably already has a reference genome. So I thought I'd also mention here, so a SNP is a variant in the context of many individuals, or occasionally just one diploid individual that has a maternal chromosome and a paternal chromosome. Like humans, we get a copy of our DNA from each parent, and occasionally parents have different variants, or sometimes what we call alleles. So here, while one parent had a G at this position, the other parent had an A at this position, and as a site in the genome, we call this a polymorphic site or a single nucleotide polymorphism. I'll also point out that oftentimes in genetics of any type, really, we frequently don't represent DNA using the double-stranded form, right? What we do is we just pick a strand, usually the plus strand as it appears in the reference genome, and we just write the DNA as one single strand. We do not, illustrate the reverse complement of that strand or the complementary opposing strand, right? So here we're just showing the maternal and the paternal sequences for one strand. So this would be what we call a heterozygote genotype. It's where a single diploid individual has two different alleles at a position. This is a homozygote here for GG, and here's a homozygote for the alternate allele AA. Okay, so what does that look like in next generation sequencing data? Well, recall with short reads, what we do is we map the reads that we've sequenced from an individual back to the genome, and we get what we call pileups. So this is a pileup of reads. Okay, so each one of these gray bars here, okay, it represents an individual read that are piled up on the genome in a position where they align to that reference genome. This is the sort of position in the genome that then varies. Gray represents here the sequence, that the sequence is identical to the reference. So the reference individual here has a C, but then anytime where in this representation an individual read differs from the reference, that base shows up as red. So you can see the T's here piled up. So in a heterozygous site, and say in a human, we would expect to see both gray and red reads because at this position because of the underlying heterozygous genotype. So this is so here you can see the actual sequence here. Here's the C down here. This is a screenshot I took from the integrated genomics viewer. This is IGV. We'll be using this in the class. I'll show you how to use it. It's a great way to visualize pileups of data to go in and check the quality of individual SNPs that may be of interest. So this is a standard whole genome resequencing workflow. We begin obviously with the sequencing of sample genomes. We then map the reads back to the reference genome, and then we typically go through a number of steps in which we try to reduce the false discovery rate. So oftentimes we get artifactual SNP calls that have a whole array of reasons why we get those artifacts. Throughout the course, maybe I'll mention some of those. But one of the things we need to do is realign around indels. So the very fast read mappers tend to not align reads around what we call indels or insertion deletions. This is a place where the reference genome has some sequence that the sample genome does not have, or vice versa, and the reads that right around the region where there are these indels frequently get misaligned. So we use more rigorous techniques right around indels to improve the alignment. We frequently will remove duplicates. So this might not seem intuitively obvious initially, but as part of the library preparation step for preparing the DNA for the sequencer, we oftentimes will PCR amplify the libraries in order to get dense enough clusters on the flow cells. But really, that can introduce artifacts because PCR makes errors in the underlying template sequence, and so we can be sequencing PCR errors, and we can detect those errors if we know that a set of reads came from the same PCR, originally PCR template. I wouldn't worry too much about that right now, but this is, I guess, the broader point here as I'm going through some of these, what might seem like details, is that SNP calling is a little bit more involved than you might expect at first, and there's a lot of steps that we have to take care to take. So I'm introducing really some of the what now I think is fairly standard protocol. So as you move on in your careers, you can come back to this lecture and some of the lectures downstream and really dig in and understand exactly why we do some of these additional steps. We frequently recalibrate the base quality scores. So these are the scores that come off of the machine from the individual reads. So these are the Phred scaled error probabilities, which I'll be telling you a little bit about in the next mini lecture. We go about recalibrating those quality scores because the algorithms that call the bases frequently overestimate just the confidence that we should have in any one nucleotide base. So all that aside, these are some details. Here's one other major point that may not seem obvious at first, the distinction between single sample calling and multi-sample calling. So occasionally you only have one sequence genome. When that's the case, you obviously do single sample SNP calling, where you align the reads of this single sample to the reference and call SNPs. However, in many resequencing applications where we have many individuals, so the 1,000 Genomes project had, I think, more than 600 deeply sequenced individuals, but the idea is to call all samples together, okay, because the presence of a SNP in one sample can influence the probability that there is a SNP in another individual at that same position. And in fact, that also helps to reduce the false discovery rate significantly. There's additional things we can do in population context to improve the SNP calling. We can use what we call linkage-based analysis to improve the genotyping. So that's really probably beyond the scope of this particular class. But again, just realize that there's been a huge community effort into developing the technology to analyze these variants and to reduce the noise associated with the data. And then finally, we almost inevitably, we almost always, have to filter the SNPs once we've run this protocol, because there are still a large number of false positives. So the reason I say we have to take care in all of this is this is another screenshot here from human alignment. We've zoomed way in here. Again, this is an IGV screenshot. Pretty cool because you can, see it right, the region that you're looking at, this red region here, right up against the genome assembly. This is chromosome 20 in humans. But we've zoomed right into individual reads here and to individual bases, so that a blue base here is a non-reference base call. But what you can see in this particular region, that you have a pileup with a lot of different base calls in a region. And the question is which of these are real and which of these are not real, and that just goes to illustrate that sometimes the SNP calling can get pretty messy. So in the end, what you end up with, this is some of my own data here from some resequencing we've done. And what I've done here is I've just reported the density of non-reference SNPs in individual samples here. So each one of these samples is a unique strain of algae. And what I've done here is to take data from all 17 chromosomes in this particular species and delimited the chromosomes by these blue lines. So the genome's about 120 megabases, or 120 million base pairs all the way across. And here are what you can see windows of non-reference SNPs, which is a really interesting pattern in this particular species. Other individuals here show sort of uniform high levels of divergence from the reference genome. So another whole genome resequencing application is structural variant detection. So SNPs is one form of variation that obviously form one significant area of next generation sequencing analysis, but another significant area is detecting sort of these chromosomal events and what we call structural variants. So there's many different classes of them. The major ones that you should probably be familiar with are like deletions, right? So here's the reference genome. The pink segment here is a segment of DNA that's present in the reference, but it's missing from the sample genome. So the green here represents the flanking sequence on either side of the reference, of this deleted section. So here's a sort of the reverse, where these are indels, right? Insertion deletions, because really insertions deletions are really mirror image of each other, and they're an insertion in one genome, but a deletion in the other genome. So this is sort of the reverse here. The pink here is an insertion in the sample genome relative to the reference genome. So it's a piece of DNA that does not exist in the reference. So insertion deletions, are most commonly 1 or 2 base pairs in length, but of course they can be very long, even hundreds of kilobases. So there's both small indels and large indels. So I'd like you to also sort of understand tandem duplications, inversions, translocations. I guess these are sort of minor differences here between tandem duplications, where a region of DNA duplicates and right next to the original copy of the sequence. Here's where a duplication sort of duplicates, or a chunk of DNA duplicates and moves to a different part of the genome. Here's an inversion where a piece of DNA that was in one orientation, say everything was pointed from left to right here in the reference, but then here you can see that this central section is now inverted relative to the reference. This is a translocation here where one piece of DNA then sort of moved to another part of the genome without leaving an original copy, as in this case. Okay, so structural variants, we'll talk a little, quite in detail about how to call these in one of the, we'll spend, I believe, an entire week working on our structural variant predictions. So this is some structural variant data that I've generated. This is what we call a Circos plot that sort of shows for 20 different sample genomes. Each pink semicircle here is a separate genome, and where you see dark hatch marks is a place where a region of the genome is deleted in one of the sample genomes relative to the reference. This is sort of the reverse here, where we have then duplications relative to the reference. Here you can see blocks of duplicated DNA sequence. Some of these are very large blocks. I believe this chunk right here spans several hundred kilobases. So these circular representations we'll hopefully become quite familiar with, and if we get a chance, I believe towards the end of the class, we'll be working to pull together some of our analysis and generate these Circos plots, because it's a real skill to be able to generate these, and it's a marketable skill, something you'll be able to tell your boss or that you know how to do. So another form of structural variant then is these translocations, and in these Circos diagrams we sometimes illustrate those translocations with sort of in this fashion, where we connect a region here that's translocated to another region, or in some cases duplicated. We can also sort of illustrate it the same way. Okay, so in many cases, though, particularly in human applications, there's not enough money to sequence the entire genome of each individual. But that doesn't mean that all should be, we should give up on genotyping an individual, because frequently what the question of interest is what disease does this individual have, or can we identify the genotypes that may make a individual susceptible to a particular disease. So the important observation here is that up to 85% of disease susceptibility mutations are in exons. And so if there was a way to sequence these exons and only exons, we'd be able to pick up roughly 85% of these mutations without sequencing the rest of the genome. So exons make up only about 1% of the genome. Okay, there's about 180,000 of them in the human genome. So by targeting a very small percentage and not wasting sequencing effort on 99% of the genome, where these mutations that affect disease are very rare, we can increase the efficiency in which we can detect mutations that are having important functional effects. So how do we go about doing this? Well, there's really, historically, there's really, I believe, 2009 maybe was the first exon capture experiment, where you design probes to the exons in the genome. So we already have the genome sequence, so we can design probes. You then pull out using various biochemical techniques, sometimes a microarray, sometimes a solution-based, sort of bead-based technology to extract or hybridize the probes to the exons in your sample genome after, of course, just shearing the DNA, hybridizing to these probes, pulling the probes out, which are now hybridized to the exons, and then eluting the exons and then sequencing them. Another more recent development now is this PCR-based target selection. So you do the polymerase chain reaction on literally tens of thousands of positions in the genome, okay, simultaneously with known targeted, primers that target exons of interest, and in some cases the entire exome, and then you sequence those PCR products. So one particular application here now, I noticed that you can order a set of primers that target exons that are known to have cancer-causing mutations, and then you can PCR using that set of primers and basically identify mutations that might cause susceptibility or risk for certain types of cancer. So this is what you would get in a typical experiment after extracting the exons from your sample. You sequence and then you read map back to the reference, and here you might expect a pileup of reads sort of like this, where individual reads match the reference here, and you get some higher depth of coverage here, whereas in the intermediate areas you don't get coverage. The stars here, I think, are meant to represent SNPs. Okay, so I'm pretty sure most of you have probably heard of this technique, the RNA-seq here. The goal here is to quantify differences in gene expression typically between two treatments. So expose a set of cells to one environment versus another, and then do RNA-seq. Sometimes you might do cell type 1 versus cell type 2. Typically we contrast two treatments, although we also do RNA-seq in cases where we want to sort of identify expressed regions of the genome. So we're not necessarily interested in just contrasting two samples, looking for differences in gene expression, but actually discovering which genes are expressed at a particular time. And in that case, you really only need one sample. Okay, so obviously we're dealing with RNA here. So at the beginning, we typically extract total RNA. I mentioned this before. We need to remove ribosomal RNA from our RNA preparations, because what we're after typically is the mRNAs, or the messages. The RNA gets sheared. We create cDNA. We sequence the short ends using single or paired end approaches, frequently just single end. We map the reads to the reference genome, and then we have to do some statistical analysis to normalize, because many times you get, if you're trying to contrast two samples, you have one, run sample 1, say, that went very well, and you have a lot of reads. Sample 2 didn't go so well, so you have maybe half the reads. So rather than inferring that sample 2 has all genes expressed at half with sample 1, or expressed at, we have to normalize the data. And we'll be doing some of this later in the class. The degree of multiplexing, in other words, how deep do you want to sequence? How many samples do you want to put on a particular lane? Really depends on how low in expression do you want to be able to quantify. So if you only want to be able to detect the very highest expressed genes, which have lots and lots of transcripts, that can be done quite easily. And you can multiplex many samples. However, if you want to detect and quantify the very rare transcripts, then you need to sequence at a much higher depth. So this is what we call the dynamic range. The dynamic range is the range of expression levels that you can quantify using a particular coverage depth strategy. Right? Okay, so then what we can do is infer differentially expressed genes using standard statistics. Again, we'll be doing some of this later in the course. So a couple of things about RNA-seq that I think you need to be aware of is one is that we can't, we're sequencing what are called mature transcripts. So these are processed RNAs. So here I'm showing you a transcript as it would appear immediately after transcription. The exons are in orange. You can always remember exons if you're not a biologist, because these are the parts that exit the nucleus, and whereas the introns are spliced out, so they're removed, and then the exons are spliced together to form the mature transcript, or what I call the processed mRNA. So this creates a problem when it comes to read mapping, right? So here is the processed mRNA, which we've sequenced. So I should mention very quickly that these introns are basically gone immediately. I mean, they are transcribed initially, but they're degraded so rapidly that we assume basically that we never detect them. And it's actually true that we really never sequence introns when we do RNA-seq. So here, if you imagine then that we've sequenced a processed mRNA where the introns in black are now missing, okay, a read would span a boundary where an intron exists in the genomic DNA. Of course, our reference genomes, like I described in the previous lecture, are really always genomic DNA, right? So when we map the reads back to the genomic DNA, and the reads that span the position where an intron used to exist or existed in the reference genome, the reads now get split. Okay, so here's the intron, here's the beginning of these reads, here's the ends of these reads. So we have to use sort of what we call splice aligners. These are sort of splice-aware alignment algorithms. You may have heard of Top Hat. It's probably the most common algorithm. So other RNA-seq applications, we can do a whole variety of things. One is to do time series analysis to infer gene regulatory networks. So we sequence RNA samples from many different time points following some exposure. We can use RNA-seq for SNP discovery. This is rare, but can be done. So in non-model organisms, say a plant that has a very huge genome, like 50 gigabases, a friend of mine is working on the ginkgo tree, and the genome is 50 gigabases in size. That's more than 15 times the size of the human genome. And really, you mostly want to be sequencing the genes. So how do you sequence the genes and discover SNPs? Well, you can sequence the RNA, and assuming that both copies of a heterozygous allele are expressed, you can detect the SNPs in the RNA. Okay, it sounds a little bit strange to not just sequence the DNA, but if you want to sequence the genes and you don't know where the genes are and you don't have a previously sequenced genome to extract the exons using exome sequencing, RNA-seq might be a reasonable application. Discover alternate splice variants, I've told you about that. Gene prediction, so in a project that we're working on, we're using RNA-seq data to tell us where the expressed parts of the genome are. We can then pipe those RNA-seq data into our SNP calling algorithm, or our gene prediction algorithms, and identify, or improve our predictions of where the genes in the genome are. There's lots of applications here. Quantify allele-specific gene expression. Yes, we can do that as well. So one application maybe you haven't heard of, this is what's called ChIP-seq, or chromatin immunoprecipitation sequencing. So the goal here is to detect the DNA binding sites of classes of proteins like transcription factors or chromatin remodeling factors that actually physically bind to the DNA. So the protein binds to the DNA, and we want to know where an individual protein binds to the DNA in this type of experiment. So it's pretty slick. What we do is we cross-link the proteins in DNA, so we physically bind the proteins in a particular sample to the DNA. We cross-link them so they're now covalently bound, so they can't sort of slip off the DNA during the course of the experiment. We then capture the protein DNA complexes using antibodies. So the antibodies are typically attached to beads. So the antibody binds to the protein part, which of course is still bound to the DNA where it was bound natively, and then we can pull the beads out and thereby pull the target protein and their complex to DNA out of the sample. We can then remove the DNA from these complexes, and then we can sequence only the DNA that was bound to the protein of interest. Okay, so we've effectively excluded all of the rest of the DNA in the genome where the protein was not bound. Okay, so this is how we can then detect small regions of the genome where proteins are bound to the DNA, right? So what we do is we take the sequences, this purified DNA, we sequence it, say on a HiSeq or other high sequencer, and then we align the reads back to the reference. And essentially what you get then is you expect to get what we call a chip peak, right? So here are the aligned reads, the pileups. I think this is coverage depth here. And what we can do is infer right at the center of the peak is where the protein is most likely to bind. So then we use some algorithm to fish out the exact sequence out of what might be a 200 base pair region where we think that the protein is most likely to bind. So this has some limitations. The resolution, is limited by the fact that when we do the short read sequencing, we shear the DNA. These fragments are generated by sonication and are typically about 200 bases, so we can't get any closer here. Usually these pieces are about 200 bases long, and also we have to have an antibody to the protein of interest, which, by the way, is a lot of work to create. So another technique here then that's commonly applied is what we call metagenomics. Metagenomics is a case where you take an environmental sample and you simply isolate DNA and you sequence the sample from the environment. So this is called environmental or community genomics sometimes. It's really a very anonymous technique, right? We don't know what organisms are in a soil sample or a sample of a marine, sort of some water we took out of the ocean, or the human gut is another place, the human mouth. These are places where we just don't know what's there and we want to know. So this is really helpful because the vast majority of these organisms can't be cultured. And really, the questions that we can answer is what organisms are there, what are they doing, and how are they doing it? So who is there? Well, decades of research in microbial genomics has provided a huge database of known samples. So these are cases where we have done lots of experiments to obtain the DNA sequence of many now well-known microbes. So we can detect them if we have that database of sequences. Because we sequence the samples, we can then identify these DNA sequences. What are they doing? Well, we start to sequence the DNA in the environmental sample, and we say detect a particular gene that's there. Many microbes have very distinct genomes with proteins that have very unique functions that do certain things, such as nitrogen fixation in a soil sample or out in the middle of the ocean. So we can, we sequence, we find a nitrogen fixing enzyme or gene corresponding to a nitrogen fixing enzyme, and that gives us some idea of what might be going on in that community. So this is very computationally intensive. It's difficult because you have incomplete linkage information, so it's difficult to know sometimes if when you get a sequence read how if it is physically linked to another sequence read. So obviously we have to do some form of de novo assembly here in order to reconstruct usable sequence. So sometimes we can do what we call a targeted approach, where you actually only sequence the samples that have a 16S molecule associated with them. So you can sequence the pieces of DNA that flank the 16S. So the 16S is a ribosomal DNA piece that codes for ribosomal RNA, and there's a huge database of known 16S sequences. So if we first pull out the 16S sequences and then only sequence the parts of the DNA that's physically linked to the 16S, in close proximity to it, then we can have a good sense of actually what organism we're sequencing. So the Sargasso Sea Genome Project and the human mouth gut microbiomes are all very heavily studied examples. So finally, today, I think I've already mentioned this a little bit, but de novo assembly falls into that category of broad stroke categories here, where we're trying to use typically paired end sequencing to sort of assemble or infer the structure of the genome. So the most basic application, of course, is in taking an unknown genome. Say the panda bear, I believe, was the very first genome that was assembled strictly using de novo assembly. And what you do is you stitch together the genome essentially by finding overlapping reads, right? Where two reads overlap, and then you extend a contake. That's what essentially these algorithms are doing, to build longer and longer and longer contigs, which eventually can be stitched into what we call scaffold, which are sets of contigs which we know are physically linked on the same chromosome, but there are gaps between them because of repetitive sequence or something that's difficult to sequence across, but we still know that they're linked, say, because one paired read is here and the other paired read is over here, so we know that they're linked because they're part of the same paired end, but we just don't know what's in between. Okay, so other cases where you need de novo assembly, not just in whole genome sequencing, but where you need to assemble regions not found in the reference genome. Okay, so we know that there are large sections of the human genome that are not found in the reference. So when you sequence an individual that has these regions, the reads don't map to anywhere. So frequently what you do is you strip out the unmapped reads after doing the alignment, and then you do de novo assembly only on the unmapped reads. And frequently what you can do in next-gen analysis then is to assemble those parts of the genome, predict genes that are on them using a separate gene prediction software, and in the end discover genes that might not be in the reference. Finally, I've mentioned this a couple of times now, we can use de novo assembly to assemble RNA-seq reads to identify alternatively spliced transcripts. Okay, well, with that, that's the end of the second lecture, so I'll see you next time.